As I explained to you before, in a clinical lab, you need to have pre-tested assays, and of course, when you've got patient DNA samples, you can buy the established tests, and there are actually 17 million SNP assays that you could buy from this company, for example. There are 1.8 million assays for copy number and 900 assays for mutation detection. So, these are all ready to go solutions. I'm going to explain to you what actually happens during a PCR reaction. So you start with the target DNA on the left-hand side, and that's on the PCR machine. It actually starts off at 95 degrees where it melts the DNA, separates the strands, and the second step, the primers start binding to the single-stranded DNA, and that's where the annealing temperature of 68 degrees, for example. And after annealing has taken place, the temperature is switched up to 72 degrees, where the Taq polymerase works optimally to extend the primers and add new building blocks, the DNTPs, to make two novel strands. And this one cycle shows an example of one molecule end up being two molecules being generated from a single molecule. So that's for one cycle. And I explained to you before that you can get at least amplification of 1 million times. So in this case, you would have to do roughly 20 cycles to get a million-fold amplification of a single molecule. So once the PCR reaction takes finished on the machine, and the scientist will start generating a report for the clinician. In this example here, it shows a SNP genotype report where the various SNPs have been amplified and a report generated, and it shows what the risk allele for each of those SNPs are in this column here. For example, the top one, it says C is if either allele has a C is risk, and what the next column shows what your actual genotype is from the test, so two T's on each chromosome, and what actually that means is that you're green or there's nothing wrong with you. In the case where you're heterozygote, there might be some effect on the phenotype. You might have some deficiency, but usually that's not the case with a lot of inherited diseases because they tend to be recessive. So again, it's highlighted in yellow here to show it might be important for a genetic counselor to advise on the passing on of a risk allele. Then the next possibility, the next outcome is to actually have two recessive mutations together. Here's two C's or two T's. Obviously that's where it's highlighted in red, so for danger. So it could basically mean that you actually identified what the genetic the molecular basis of the disease might be. And just to point out that in most of these cases, you actually have an internal control that you use to validate to standardize your tests in an established assay. I just want to go back to the original slide. Yeah, I want to recap on the whole idea of why we're using clinical genomics. It is to identify the DNA sequences, how they've changed and caused the molecular mechanism. And in the previous report, you saw the red. So just to remind you why we were doing that. So that was an example of sort of an established clinical assay and getting a report from it. In this one, this brings you to the current status of actually using the PCR to detect coronavirus. And unfortunately, there's no assay as such for coronavirus because it's a new virus. So what you might get in a clinical report is more like an R&D a report where you have to, the scientist and the clinician really have to sit down and work out what's going on, because the results aren't very clear from some of the things. PCR is a very reliable process, but the problem is whether you're actually getting enough DNA in your sample, for example. This is sputum, which is basically excretions from your cells in your nose. So it doesn't actually have any live cells as such. So as a result, it becomes a bit difficult to find out if your sample in fact is truly representative of what the infection particles are, because it doesn't measure internal virus particles. So this is a bit of a problem in developing a foolproof assay with PCR, but I'm sure in time to come this will become more standardized with new information coming on board. But just to emphasize that the PCR test used for the COVID-19 is still experimental. That's it. Thank you.